receptors are cleared from the circulation with half-lives that approximate those of small molecules. So you have to take that into consideration. In addition, the volume of distribution of a typical antibody is limited to the plasma compartment. In this case, the volume of distribution is much greater, and you have to take those PK parameters into effect as well. And that can have practical implications on, is the target you're going after feasible from a manufacturing and a commercial standpoint. So as I mentioned, with antibodies, you have long half-lives, and that gets around the fact that you have to administer these through IV infusion. So not as convenient as taking a pill at home, but nonetheless, it doesn't prevent antibodies from, hasn't prevented antibodies from not only being very effective treatments for patients who suffer from diseases, but also being very big commercial successes. When you take a targeted delivery approach, you negate some of the PK advantages of the antibody naturally. And this will require more frequent dosing. And that more frequent dosing will depend on your target ligand stoichiometry and how long your target needs to be inhibited or agonized to give you a pharmacological outcome. And then beyond that, the pharmacodynamic effect of that. So in the case of some GPCRs, perhaps, you may be able to have a much longer pharmacodynamic outcome after a single dose, so you may not need to dose as frequently. But in the case of BASE, where we had a very nice talk just preceded mine, you've got to have the enzyme inhibited on a relatively regular basis. This is going to require quite frequent dosing. In addition, a bispecific molecule is a very challenging from the concept, from the perspective of scalable manufacture. Genentech and other people have recently described some very ingenious solutions to this. And if you go that route, certainly access to those licenses or partnering your idea to those companies will be a path forward. Cost of goods. Take into consideration the human equivalent dose for, if it worked at 25 mg per kg, it's going to be about 1.25 gram per 70 kilogram person. By example, two antibodies I pulled out of the literature, Tisabri, our own antibody, which has to be dosed at relatively high levels to maintain receptor saturation, is dosed at 300 mg IV every 4 weeks. Humira, an anti-TNF antibody, depending on indication, is dosed in a range of 40 to 160 mg IV weekly or once every 4 weeks, depending on the indication. So you can see there's a reasonable difference in the amount of product you'd need to get on board to have an effect with an anti-base antibody, even if it is targeted delivery. Then you factor in all the aspects of whether you're going to do monthly dosing or bi-monthly dosing. If you want to do a 300-person-year phase 1 clinical trial, that's about 12.6 kilograms of drug product you have to get. So it's a reasonable demand on manufacturing. So that's where I'd like to switch over for the conclusion of my talk, and I apologize I'm running over here. So I'm going to go real fast through some of the CMC considerations, timeline to IND, and cost. First of all, I just want a quick illustration of antibody, the growth of antibody therapeutics across the industry, as documented in this review by Nelson et al. That came out in Nature Reviews Drug Discovery. And they illustrate very nicely that relative to the decade from 1990 through 99 versus 2008, there is a decrease in the number of murine or chimeric antibodies, and a progressive increase in the number of humanized antibodies to human antibodies entering clinical trials. And here's a plot of the increase in number of antibodies going into clinical trials. The majority of those are going in for naturally cancer and inflammatory indications, and very few right now at this current stage, of course, into CNS. But the other point I want, this illustrates very nicely, is the success rate with biologics compared to small molecules. We've heard and are well aware of that. And they show here in transitions from phase 1 to phase 2, phase 2 to phase 3, and phase 3 to phase 4, the percent of antibodies that have successfully moved forward. And you can see that there's a very high success rate for both humanized as well as human antibodies, or coming from different other technologies such as phage display. So some of the things, the upfront considerations of biological therapeutics is that, of course, they offer you the advantages of excellent target selectivity, affinity, a very predictable PK, metabolism, elimination. You do not need to follow metabolites or extensively characterize metabolites. It's a much more straightforward path for IND-enabling studies relative to small molecules. The other consideration is if your antibody is cross-reactive with the antigen in both humans and your preclinical model, it's a straight path to development. You can either, if it's a mouse antibody, you can humanize it, or if it's a mouse antibody, you can get a human version through appropriate phage display type of technologies. If you're neutralizing antibody that works in a preclinical model, does not cross-react to the human antigen, now you've got a little bit of work to do because you've got to find another antibody that's equivalent to your active antibody that cross-reacts to the human and demonstrate bioequivalence of those. So that's a little bit of work coming out in there. And then ultimately you want to confirm the activity of the humanized antibody in a preclinical model of disease. Primates typically do not have the sorts of diseases that we can test conveniently. So you typically go to rodents. And there you can, it's enough to provide evidence of target engagement through acute situations. And if you really want to do a chronic treatment paradigm, you can induce tolerance in mouse models to show that your humanized antibody has the same activity. Real quick word about the different antibody technologies, and this could be 3 or 4 talks easily in a focus, but the 3 principal platforms are humanization, phage display, or transgenic mice. And between humanized versus human, the trend is going to fully human antibodies, with just the thinking that they're slightly less immunogenic, but to date there is no solid clinical evidence that fully human antibodies are any more superior than humanized antibodies from the consideration of immunogenicity. Just quickly, I'm going to show you what humanization involves, but the main point I'm going to get across here is that unlike the other 2 technology platforms, phage display and human transgenic mice, humanized technology is approaching the end of its patent life cycle. Most of the primary patents are going to expire over the next 2 to 4 years. So it's going to be a generic technology. And it's a very straightforward technology, just for the benefit of anyone who's wondering what the heck is humanization. It really starts with the mouse hybridoma that's expressing a mouse antibody from that's target-specific antigen. You can easily clone out the variable regions through PCR technology, generate recombinant constructs that express a chimeric antibody. Through sequence analysis of the variable regions and alignment to basically sequence informatics and molecular modeling, you can design a human equivalent at the primary structural level of your mouse antibody. Initially, they used to be CDR grafted, but more refined techniques involve a little bit more detail than that. And then reverse translation of that and popping it back into a vector. And that's how you generate a humanized antibody. Some other details regarding small molecules versus biologics real quickly. I'm not going to get into terminology here, but small molecule, you have customized manufacturing. Each molecule is different. It's going to require its own process development. Biologics, by and large, antibodies in particular, is a platform process. It involves a very straightforward recombinant DNA-based approach to generate the composition of matter. And then from there, cell-based production and scalable bioreactors. And process improvements now are giving titers for production on the order of grams per liter. So there's been extensive progress in manufacture of biologics. There are an extensive series of QA and QC quality control tests for the biologic. And another aspect of biologics is the timeline it takes to get material to get yourself into the clinic. This is a standard sort of boilerplate timeline of about 18 months to get IND material. It can be contracted to as little as 15 months, depending on where you go. But the steps involve basically generating your vector, generating cell lines, finding high-expressing clones, developing, ensuring their characteristics for production in bioreactors, and then working out the processes to purify the antibodies, and they're really very straightforward. Of course, protein A and some other polishing step, and then at the end of the day, formulation, stability testing to filling the final product. And this is where the costs come in, and this is where, of course, if you're doing this as an academic lab, here's where you need to partner, because manufacturing up to the point of cell line development is about $1.5 million. The drug substance manufacturing from bioreactors, stepping up from 10, 20 liter to 80 liter to thousands of liter bioreactors, can run around 6.2 million. And the final fill finish of the drug manufactured product, stability testing, is close to another $1 million. So that's where the big money starts coming in terms of manufacture of biologics. And for my final slide, I, not being a student of Latin at any point, I originally had titled this entrepreneur and tour, and I thought about this and I thought, no, it might be caveat entrepreneur. So anyways, bottom line is I really love this quote that was attributed to Michael Green, the former CEO of Geron, commenting in an article just last year in Nature about Geron abandoning stem cell therapies. And the final thing he had to say was, the first one out the door gets all the arrows in his back. So if you're going to try something new, be prepared for that, develop a thick skin, but don't give up on it. The other aspect that's really illustrative is from a review by Steinmetz and Smart about the cost of drug development. It doesn't matter if you're in small molecules or biologics. It's a money pit, as you can see. From the discovery phase, which is usually funded through NIH and foundations such as ADDF, to early preclinical development and early clinical trials, you're burning cash until you get to the phase, late stage clinical phase. If your drug is unsuccessful, of course, you're out everything you put in. And that's why drug companies are so very careful to take on big projects, and they have to have good evidence to take on this amount of expenditure. But if success starts to come up, then somewhere shortly after commercialization is when you start recouping your investments. And if the drug is unsuccessful as it approaches market, you get a little bit of money, but depending on the degree of success of the drug is where the payoff comes in. So just to summarize, there are opportunities in neurodegenerative disease for biologics, as I've tried to illustrate for you. Apply and exercise principles of sound drug discovery as you progress your idea. Targeted delivery is a great idea. It does pose challenges from the perspective of drug development, and you should be aware of that and cognizant of that. And at the end of the day, antibody therapeutics offer a robust, scalable platform with a proven market value at a cost. Thank you.